for us, sometimes the most difficult part is how can we model the human phenomenon? So at that point, we felt like we had a good model to study the system. And then, reading the human literature, we found that there was evidence that one part of the brain, particularly the cingulate cortex, was active during the placebo analgesia task. So we used a number of techniques, but one of them is to use miniature endoscopes that can collect fluorescent signals inside the brain. It's called a miniscope. It's been invented by Mark Schnitzer at Stanford.
So you literally mean like a miniature microscope that is hooked up to the animal's head.
Yeah, so it's tiny. It's about 3 grams. And so you can mount this on the mouse head like a hat. And the mouse, because it's slide, mice behave normally. They function completely normally. And so what you can do with this is that you can then use viruses to express in certain neurons that you want to study a calcium indicator. And so when neurons fire action potential, the intracellular concentration of calcium is going to rise, and that's going to change the fluorescent signal that you can then record at the single cell level with this microendoscope that we call a miniscope.
I see. So you can engineer mice such that specific neurons in the brain, in the amygdala, in the cortex somewhere, basically wherever you want, you can make those neurons literally light up if they are sending out nerve impulses or action potentials. And then because you've got this endoscope, this miniature microscope hooked up to the animal's brain, you can actually record when those neurons light up